can send or upload the video somewhere for a few hours or days.
Great, thank you.
Okay, so yeah, so I will start the recording now. And I see we have some more people that already arrived. Okay, so oh, it says stop recording. So we are already recording. I think somebody from our class already started it. Yeah, it says stop recording, so it's being recorded. Okay, so let's start then. This is the last Brno Bioinformatics seminar from the Faculty of Informatics, and our guest today is the second guest from Kiev, Ukraine, Dmitro Horislavets.
Yeah, that's right.
He will talk about working with the Bruijn graphs and sequences in a special way. I believe he did the talk will be based on things he did during his summer internship in Switzerland at the Swiss Technical Institute in Zurich. He's now back in Ukraine, and today they had another barrage of missiles coming from Russia. So I think he told me he will switch off the camera later on, but now you can see him. So the floor is yours, Dmitro. And if I forgot to something in the introduction, please complete it, and welcome.
Yeah, thank you, Matej. So I'm starting to share in my screen. Okay, so hello everyone. My name is Dmitro. I'm happy to be here. Thank you all for the opportunity to give a talk. I'm studying for a master's degree in computer science at Kiev Academic University. My mentors are Alina Frolova from Institute of Molecular Biology and Genetics from Kiev, and Andrei and Harun from ETH Zurich. Today I will tell about the applications of the Bruijn graphs, in particular in metagenomics. So I'll start with some biological preliminaries, some basic stuff. So we know that living organisms are built of cells. The cells contain chromosomes, and the chromosomes consist of DNA, where DNA is a double-stranded polymer molecule. It is composed of units called nucleotides. Each nucleotide is composed in turn of one of 4 types nuclear bases, a sugar and a phosphate group, and the sequence of these nuclear bases encodes genetic information. We also know that among us there are other life forms at the Earth, and some of them directly affect our life and health. I mean, different bacteria and viruses, and it is reasonable to study those microorganisms on a genomic level in order to understand better the nature of diseases that are caused by them. But there is a problem that the vast majority of bacteria cannot be actually cultivated, because as they are collected and they appear in laboratory conditions, they enter the so-called viable but not culturable state. It is believed that it is their response to the environmental stress. They remain alive in this state, but they cannot be cultivated. That means that the only way to analyze those microorganisms is to collect them directly from the environment. And that's where metagenomics comes into play. So metagenomics is the study of the structure and function of entire nucleotide sequences, isolated and analyzed from all the organisms in a bulk sample. And this sample can be collected from different types of environments. This can be either, I don't know, sewage or, for example, human skin microbiome or soil microbiome, depending on what you are studying. Here you can see that typical pipeline of metagenomics. So the first step is to collect DNA, to extract DNA, then those strands should be fragmented, sequenced, which result in a big number of relatively short sequences, which can be later used for different purposes, depending on the type of study that you are conducting. Here are some applications of metagenomics. It can be used to monitor environmental pollution, it can be used to investigate some soil microbiomes, to provide some profit for agriculture. It can be used, as I said previously, in medical field, and there is an important study of also of genes that help bacteria to be resistant to antibiotics. I would like to take a closer look on the sequencing step. And here on the left-hand side, you can see very popular method of sequencing, which is called Illumina sequencing. It consists of the step of collecting DNA, fragmentation. After that, the adapters are added to the ends of those strands so they can bind to flow cell. This step is followed by amplification step. After the clusters are formed, they are sequenced by adding nucleotides with special fluorescent adapters, so that each nuclear base has its own color. This method actually results in, as I said also before, a very big number of relatively short sequences. And that's why the concept of K-mers is actually widely used in computational biology. Here on the right-hand side, you can see the example of a 3-mer. So K-mer is basically a substring of a string of length K. So here in the string ATGG, there are two 3-mers, ATG and TGG. They are used to, while we get after sequencing those lots of DNA sequences, they are split it in those K-mers in order to operate more efficiently, because operating with a fixed size K-mers is easier than operating with those resultant sequences. Also, operating with a set of these K-mers is faster and easier than with those sequences. So the K-mer approach is widely used in lots of algorithms. Also, as the sequencing cost decreases, the amount and capacity for sequencing data shows exponential growth. This creates a need for indexing of and searching this sequencing data at a peta-based scale, where peta means 10 to the power of 15. Just for comparison, the approximate human genome length, entire human genome length, is about 3 billion base pairs. So that's a scale of 10 to the power of 9. So that means that there is really a large amount of data. And one of the promising approach to meet these demands is to use compressed K-mer graphs. Now we came to the most important object of this talk. It is the Bruijn graph. The Bruijn graph is defined as a directed graph of overlaps between sequences of symbols, and the colored Bruijn graph is a generalization to distinguish multiple sample. If you take a look the graph on the right-hand side, you can take some two adjacent nodes. For example, let's take this one, and the graph is defined in a way that it must overlap with its direct successor by two symbols. Specifically, the suffix of length 2 here is exactly the same as the prefix of length 2 here. And of course, the length of these K-mers inside of the graph nodes differs. It's adjustable parameter. In this example, the graph is so-called 3-dimensional, meaning that length of those K-mers is 3. So the colors are added in order to store one graph for different sequences with different labels. I mean, we could store 3 separate graphs for these 3 sequences in this example, but why if we can store them in one graph and just color the corresponding nodes in the corresponding colors? This means that, for example, this one K-mer is present in all of these 3 sequences, here, here, and here. And this one is present only in the green sequence. It's very simple to understand. And accounting the Bruijn graph is a notion that generalizes annotated in each node label relation with some additional attributes. Those attributes are typically K-mer counts, which means the number of K-mer occurrence in the sequence, or K-mer coordinates, which means K-mer positions in this sequence. So now I would like to tell you about the metagraph framework. It is a scalable framework for DNA assembly, search, and alignment. It is created by my mentors, Andre from ETH Zurich team. And it efficiently uses the Bruijn graph, and it is available to store K-mer sets on a petabyte scale. So here is the concept, how it works. It's also pretty much self-explanatory, but still let's take a look. So we have samples, the separate graphs are built, there's the optional step of graphs cleaning, and then those graphs are united in the one graph with labels, which is stored in a compressed format. Now I would like to tell you about the compressed format in which metagraph stores the K-mer index and its annotations. So here are also some possible queries against the graph. So here you can see how with this query you can calculate the matching with the initial sequences, or to attempt to do some alignment to this graph. So let's start with storing the K-mer sets. Metagraph employs several data structures for storing them. This can be a simple hash table, a compressed indicator vector represented as bitmap. It has length of 4 to the power of k, indicating which K-mers are present in the set. And the most interesting one is the succinct Boss table. It is named after the first letter. This format is named after the first letters of the surnames of its authors. And let's take a closer look on this third type of storing the K-mer sets. But first, I would like to elaborate a little bit on the term succinct. The succinct data structure is a data structure that uses an amount of space that is close to the theoretical minimum, to put it simply. And the most popular approach to build the succinct data structure is to use rank select dictionaries. That means that those operations are used along with some bit vectors. Those operations are very simple. They're defined here. So, for example, for some string S, the operation rank for symbol C, and the position I is defined as the number of occurrences of symbol C among the first I symbols in the S. So the string S contains symbols from this alphabet sigma. And the operation select returns the position in S of the jth occurrence of symbol C. So let's return to the Boss format, and I would like to tell you how it is built. So consider this graph example, the Bruijn graph example. The $signs are just padding to ensure that each node has incoming and outgoing edges. So the first step to build the boss table is to take each node and edge pair and sort them into in collects order. Collects order means that these labels are sorted in reverse alphabetically. So by their last symbols, they are sorted in alphabetical order. And these edges are outgoing edges of these nodes. You may notice that some edges are marked with minus symbol. That is done to disambiguate the edges that the same incoming edges to the node. So for example, for the node ACG, there are two different edges, but with the same label. So in order to distinguish them, one of them is marked with minus size symbol. After that, the array L is included, which is the array that indicates unique nodes, because similarly for the incoming nodes, incoming edges, node can have several outgoing edges, and in order to like mark the unique nodes, only one of them is marked with the one value, and the other one have the value of 0. So in this table, it is done just by setting the 0 bits to all the nodes with similar labels, except the last one. Only the last one has the value 1. That means that nodes with value 1 from this array are unique. The next moment is that there is no actually need to store these node labels completely. There is only a need to store their last columns. And to be more precisely, there is also no need to store these last columns because it is sorted already in alphabetical order. So there is only need to store the start positions of these symbols. This is done by using this array F. And basically that's all. So the boss representation consists only of these three components: the array F, the array L, and the array W. And the array node with node labels is not stored, it's just written here, depicted here just for clarity. That means that all these three arrays together provide support for a full suite of navigation operations. Let's take a look, for example, how an operation forward can be implemented with the operations rank select on these three arrays. But first, let's notice such thing that node labels maintain the same relative order as the edge labels. What does it mean? It means that since the edge is actually defined by its incoming edge, and the edge is represented in the array W as well as in the array F. Due to this, the same relative order is preserved for these edges. So following an edge, in this case, is simply just finding the corresponding relatively positioned node. So let's take a look on some example. For example, with these tables that we have, let's say we would like to find the node to which point the edge with index 2. So we start from this node and we just want to go forward with the operations rank select. The first step is to find the rank of this symbol C in the array W. Here it is 2. The second step is to find the starting position of this symbol in this sorted array. I'll recall that the node array with node labels is not stored explicitly. Only F, L, and W are stored. So as we have rank of the C, or rank of our symbol, and the starting position of our symbol, we find just relatively positioned node by selecting the fifth position in the L array. So it's just 3 plus 2, so we land here. We can make sure that this actually works correctly because this node with this edge to obtain the its direct successor, we have to shift this label to the one symbol to the left by one symbol and obtain TA and add the C symbol to the right and get TAC. So yeah, that works. Similarly, backward operation is done, but just in different order. So if you'd like to get the direct predecessor of this node right here at the position 5, the first step now is to find the starting position of the C in F. Yeah, and we know that the last symbol of this node is C because this can be derived from the F array. If we want to find the previous node for the node with index 5, that means that it is in this range, 3 to 7. So we know that the last symbol is actually C. So the starting position of C is 3. The second step is to find this starting base from which we would like to start, and the rank to the current edge right here is 5. No, we just calculate the rank of these symbols at the index we started from. So once again, we would like to find the relatively positioned edge. So and get the second C in the W array. So we select just the second C in this array. And that gives us the previous node of this node. So these operations are done in constant time. And they can be used to implement some more complex operations. So for example, out degree, in degree, which returns the number of outgoing or incoming edges to the node, or to follow the edge labeled by some specific symbol. So that means that within this data structure, you can easily navigate through the graph, and it is like a compressed data structure, so it's more efficient than storing the completely graph, for example, in adjacency lists. Now let's move on to the annotations. I'll remind that annotation attributes can be k-mer counts or k-mer coordinates. Counts are the number of occurrences of this particular k-mer in the sequence, and k-mer coordinates are the positions of this k-mer in this sequence. But first, let's take a look how a binary relation can be encoded, because it will later appear while compressing the sparse matrices. So at the left-hand side, you can see the binary relation. Right here is like a very simple concept. And on the right-hand side, you can see the data structure called veiled tree. It is actually a data structure used to store strings in a compressed space. It generalizes the rank and select operations that are defined on bit vectors to arbitrary alphabets. So it's also not very complicated. It is also with an example here. So for example, this string E, H, D, H, E, A, and so on, how to encode it using the veiled tree structure, data structure, is to split the alphabet in 2 parts at the first step, from A to D symbols, and the range from E to H symbols. That creates a partition of these symbols based on to which part of the alphabet they belong. And the process just repeats at each step until the alphabet partitioning reaches its limit. So to encode the binary relation with veiled tree, there is a concept called BRWT. So it's just binary relation veiled tree, and it's like very similar to the encoding of strings. Once again, if we have a binary relation, just simply the alphabet is being split in 2 parts. and the elements are sorted depending on which part they belong to. So, for example, this element 1 in previous example, let me just jump backwards. 1 has values only in E and H rows, so it belongs only to this part of the alphabet, so it goes right here. And the same is being done for all other indexes. The one more extension of the basic BRWT is a multi-BRWT. This is just an extension that allows its nodes of this data structure to have arbitrary number of children, and as well as to arbitrarily distribute the columns of this matrix to its children. I will not get into details of this data structure. I will just say that it shows some improvement in compression performance over the basic BRWT, and it is actually used in the metagraph to compress those binary relations. And now we get to the compressing of the attributes matrices. So here at the panel A, you can see the general scheme how to compress the sparse matrix of attributes. It is done by using an indicator matrix which is stored in multi-BRWT format and a corresponding array of attributes. At the panel B, you can see how you can retrieve the value of this column, this attribute for some certain k-mer. With this representation is just simply using the rank operation on the corresponding array of attributes with this indicator column. And if we deal with some sets of elements for each k-mer, so for example, this can be k-mer coordinates, as I said before. So that means that this k-mer, for example, appears at the positions 2, 6, and 10 in the original sequence, and so on. To encode this case, just the additional delimiters array is included, just to separate these different arrays of coordinates for different k-mers. So it's also very natural, and it is done with the select and rank operations. There is also a peculiar feature that can be used to additionally compress those k-mer attribute set for the coordinates case. So let's take a look on this example. And there is a sequence which is split into this set of k-mers, and we can notice that the adjacent k-mers in the graph tend to have similar sets of coordinates. This is just because they are located near each other in the original sequence. Specifically, the coordinates of the successor for some node are typically increased by 1 with comparison to the coordinates of the start node. And this feature can be used to additionally compress these coordinates so that we would store not these coordinates, but some type of their differences between each other. That is done in a following way. Some of the nodes are selected as anchor nodes, and the compression is not applied to them. They are needed for compression and decompression of the sets of attributes for other nodes. So in order to encode the attributes array or set for this particular k-mer, the operation of different symmetric set difference is used. So the first step is to add 1 to all of the coordinates in this set. So it becomes 5,9, and the symmetric set difference with its successor is done in order to obtain a compressed value of their attributes, set of their attributes. Here it is like an empty set because 5,9 symmetric set difference with the 5,9 set results in an empty set. This just basically means that every time the AGC chimer appeared in the original sequence, it is followed by the GCT chimer. To reduce this information, this can be used in compression. The same is applied to all of the other nodes. And we can see that those red sets are indeed smaller than the gray sets. Of course, this operation is invertible, and it's just based on properties of the symmetric set difference operation. These sets of coordinates are also easy to decompress back, of course, with the use of anchor nodes. Here are some formulas for that. So now I would like to tell you about my current work with the metagraph. I am working right now on problem of returning the set of all input read sequences that overlap the query with from the annotated count in the Bruijn graph. I started to work on this during my summer internship, and I'm continuing working on that problem, which will result in my master thesis. This like query and algorithm can be useful for further downstream analysis after search. I will elaborate on this a little bit later with some use cases, and also it can be also helpful during the graph assembly step to provide more refined graph cleaning. So now I would like to tell you how I realized this algorithm for now. It's, to be honest, like kind of brute force and is inefficient, but this implementation helped me to get familiar with this framework, and it set the ground for further work in this area. So for now, it works as follows. Consider this example, one more example of the Bruijn graph, and the input sequence ATGC. So I'll remind that the task is to return all these sequences from which the graph was built that contain this sequence, this input sequence. So the first step is traversal of the graph, the complete traversal of the graph actually, that results in like the set of all visited nodes, which makes available to fetch their annotations, which are stored in a compressed format, as well as the graph itself is stored in a compressed format, and that's why the traversal is being done. So the second step is to fetch those annotations, and as we have both the graph and annotations, the start and the initial and terminal nodes for these sequences are determined, and the like walks in this graph are reconstructed. So you can see that indeed this sequence is present in these sequences. What's wrong with this approach is that the graph is being traversed completely, and if we deal with very large graphs, this is highly inefficient to traverse them completely. That's why the further works implies improvements in scalability. That means that we would like to implement local graph decompression, which means that the graph will be traversed like sequentially, and the annotations will also be like decompressed sequentially without the full traversal of the graph. Also, there is a desire to implement a batch mode, which means that several queries will be processed at the same time. Here are some use cases. It can be used in environmental metagenomics, as I said, for search of unknown DNA, for example, for taxonomic profiling, because we have like a data set of collected by a consortium metasub. That's a international consortium to which my mentor Alina belongs to, and they collected a large amount of samples, and the problem there is that lots of DNA is actually unknown. So this like feature of the metagraph can possibly maybe give some interesting results by querying those sequences in some basis. And also here is mentioned a single cell sequencing. That is also a possible use case. So like to search the transcript in the graph that contains that is built of different cell types. So that's it. Thank you for your attention. I will be happy to answer your questions if you have some.
Yes, thank you very much, Dmitro. Very interesting, exciting research that you showed here. Personally, for me, a lot of this was new material, so it was really interesting. So let's see if anybody of the people online or in the room have any questions. Oops, doesn't happen. Okay, I have one question in terms of practical use. I mean, our semester was about reproducibility and scalability. So your lecture was a very nice finish for our semester, because you had scalability on one of the slides even. And so my question is about scalability, like in practical situations. How quickly does the time to search the graph grow with the increased number of sequences, or which kind of sequences are more difficult or easier to search or store, and so on?
Well, I'm not sure if I can properly answer your questions, but as I know, these graphs are typically very large, so they are at petabase scale. And I'm not sure if some sequences are harder to search or like some easier to search, some are easier to search. I'm not sure about that. So yeah, that's it. The main idea is that this approach isn't good for in general for the search and for the search in the graph, the sequence, because the full traversal is bad. And as for the different types of sequences, I'm really not sure if there are better or worse sequences to search in graph.
And did I understand properly that you store the graph representation in this BOSS format, and then you store the annotation separately in the in that binary relation format?
Yeah, sure. And that is basically the reason why the graph is traversed, because if we had the graph in some other format, then the task will not be like difficult, or it will not be even a task, because it wouldn't be even a task, because just all information will lay on the top, so we can just see or look up which sequences contain this particular query. But when they are stored in a compressed format, then in order to get the successor nodes for the nodes and their predecessor nodes, and to fetch the annotations, the graph should be traversed.
Any questions?
Maybe I have one.
Yeah, there is a question in the room.
Isn't there a loss of information when you have some duplicities?
Could you hear the question?
Very badly. Would you mind repeating it, please?
Yeah, that if there are some duplicate sequences stored, isn't there a loss of information? That was the question.
Do you mean like the completely the same sequences are stored in the same graph or something like that? Or
if there are some duplicities in the sequence and store them in the graph,
well, that will basically result.
Sorry.
Yeah, it's good. I just think I understood your question. So if you mean that like the sequence is highly repetitive, so it can consist of the same chimera which just repeat constantly, yeah? You did mean that?
Yeah, or two different sequences or one sequence, yes.
Yeah, so well, the graph in this case will be constructed of like a few number of nodes, and the sequence like order will be stored in the annotations sections with the chimera coordinates. So if you have a highly repetitive sequence, then the graph will be like small, but the annotations like but the annotations with chimera coordinates will just have those relatively large sets of coordinates, which will just tell you on which position this chimera is located. So it will appear here in the annotations matrices, and the graph will have a small number of nodes.
Yeah, so there is no loss of information, but the graph doesn't grow with the repetition, only the annotations do.
Yeah.
Mm-hmm. And maybe the last question from me, this Metagraph software, is it available somewhere, or how, if we wanted to test it, what should we do?
Yeah, of course, it's an open source, and I included here a link to the website, and there is also a GitHub repository. Okay, let's open it right now. And yeah, that's it is available to like test, to run, and so on.
Yep, thank you. Okay, so if there are no more questions, let's thank the speaker one more time. And we will have another round of bioinformatics seminar next year. So just those of you who are interested, keep watching the website. And thank you again, Dmitro, and also Alina for arranging this. And we hope to see you again, maybe at one of the other seminars we will have next year.
Thank you for having us.
You're welcome.
Yeah, thank you very much for your attention. That was a great experience.
Okay, and then the recording, through email, I will let you know where you can get it. Okay, bye-bye.
Bye, everyone.
Bye.